This is the presentation on Taxol by Mary Rexroth and Mackenzie Sterling. Taxol is arguably the most successful anti-cancer drug of all time. Taxol, generic name Paxlotel, is a microtubule stabilizing drug that is approved by the FDA for the treatment of ovarian, breast, and lung cancer, as well as a form of sarcoma. It is also used off-label to treat several other types of cancers, including cervical, prostate, head and neck cancers, in addition to lymphoma and leukemia. Like many other chemotherapies, Taxol also has its side effects. They include hair loss, hypersensitivity to reactions, nerve damage, and bone marrow depression. Most of us probably watching this already know the process of tumor and cancer development, but as a short review to understand what Taxol does, we should look at how cancer occurs. So the fundamental abnormality resulting in the development of cancer is the continual unregulated proliferation of cancer cells. Rather than responding appropriately to the signals that control normal cell behavior, cancer cells grow and divide in an uncontrolled manner. One of the fundamental features of cancer is tumor clonality, the development of tumors from single cells that begin to proliferate abnormally. As seen in this picture, you can kind of get a visualization of what cancer does. In these cells, we're seeing that they develop from a single initially altered cell and then continue to divide and create more cells that are abnormal. Specifically, how Taxol kills cancers is not entirely agreed upon. Taxol has long been recognized to induce mitotic arrest, which leads to cell death in a subset of the arrested population. It has been the dominant perception that the anti-tumor effects of Taxol occur due to its ability to arrest cells in metaphase on bipolar spindles. So in other words, it stops the uncontrolled cell divisions of cancers by forming extremely stable and non-functional microtubules. However, recent evidence demonstrates that concentrations of Paxlotel are too low to cause mitotic arrest and result instead in the multipolar divisions, cells entering mitosis in the presence of concentrations of Paxlotel equivalent to those in human breast tumors form abnormal spindles that contain additional spindle poles. This is seen in the image above. Rather than mounting a long-term mitotic arrest, these cells enter anaphase and divide their chromosomes in multiple directions.
As you'll see when we get to its synthesis, Taxol is an isoprenoid, which we just learned about in class. It is complex, which involves 19 steps and 11 stereocenters. Some general background information on Taxol is that between 1960 and 1981, the National Cancer Institute and the U.S. Department of Agriculture collaborated on a plant screening program that collected and tested 115,000 extracts from 15,000 species of plants to identify naturally occurring compounds with anti-cancer activity. Samples from a single Pacific yew tree, Taxus brevifolia, were obtained by USDA botanist Arthur Barkley on the last day of his expedition in 1962. After his return, crude extracts from bark, twigs, needles, and fruit were tested, and bark extract was found to be cytotoxic. Taxol showed mixed results in preclinical trials and was not uniformly considered the most promising plant product. Several clinical trials were delayed because of a shortage of Taxol, the only source of which at that time was the slow-growing T. brevifolia. After more than 30 years of research, Taxol was approved for the treatment of non-small cell lung cancer in 1999.
The first part of this pathway begins with the MEP pathway, which is the plastidile route from pyruvate and glyceraldehyde 3-phosphate via the methyl phosphate. on top we have isopentyl diphosphate and the bottom dimethylallyl diphosphate, both metabolites that we are familiar with. So the way in which these two join together is seen on the left here, where dimethylallyl diphosphate loses its oxygen into phosphates here, and then that double bond there is able to resonance stabilize in order to bond with the isopentyl diphosphate. These two continue to be added until they form the geranyl diphosphate molecule via the geranyl diphosphate synthase. After this 20 carbon precursor is made, a series of 19 enzymatic steps yields Taxol.
To further understand Taxol biosynthesis, I'm going to explain the abbreviations used in the pathway. PAM stands for phenylalanine aminomutase, which is shown in the box at the bottom of the slide, right there. OPP, which we are familiar with, denotes the diphosphate moiety. AC and BZ denote acetyl and benzoyl groups, respectively. So, to further show the synthesis, the first step after the geranyl diphosphate formation is the committed step. It involves the cyclization of GGPP to taxodiene by plastidyltaxodiene synthase, which is shown in detail on the next slide. So that forms that molecule right there. Following taxodiene synthase, several hydroxylation and acylation reactions occur, resulting in a hypothetical intermediate right there. Because the relative order of C1 hydroxylation and oxetine formation is uncertain, the hypothetical intermediate illustrated could be at the level of an acylated hexol rather than a hepatol bearing the C1 hydroxyl. The formation of the intermediate is then followed by an oxetane formation and C9 oxidation, forming baccatin 3, right there. After the oxetane is formed and C9 is oxidized, the side chain, beta-phenylalanine, which is formed by PAM, right there, is then added to baccatin 3. This formation yields beta-phenylalanyl baccatin 3, which undergoes hydroxylation and benzylation to form Taxol.
As stated previously, taxodiene synthase is the first committed step involving the cyclization of GGPP, geranyl diphosphate. So this process begins by the cyclization of geranyl diphosphate by taxodiene synthase with the ionization of the diphosphate with the closure of the first ring seen here. So as we can see, these electrons attach here and here, and this leaves. Then there's a unique proton migration from C11 to the ray face of C7 here. And then that leads to the closure of the second ring here, the carbocation. Next, we see a deprotonation, probably with a base, as we know how to do, like that, with a resulting cation from here. to form the major and the minor product here and here.
As mentioned earlier in the presentation, the synthesis of Taxol involves about 19 enzymes. The second enzyme we are going to talk about is taxodiene 5-alpha hydroxylase. It is the first oxygenase of the Taxol pathway and immediately follows taxodiene synthase using its products taxa-4,20,11,12-diene and taxa-4,5,11,12-diene. Fun fact, taxa-4,20,11,12-diene was first proposed to be the committed precursor of taxoid biosynthesis in 1966 by scientist B. Liggho when Taxol was still in the beginning stages of its discovery and research. So the pathway of this enzyme starts with deprotonating from one of two positions on the isomers, depending on which one, to form an allylic radical intermediate that is resonance stabilized. For taxa-4,5,11,12-diene, it attacks this hydrogen right there, which attacks back to form the radical. But if we're talking about taxa-4,20,11,12-diene, it attacks this hydrogen, which then forms the radical as well. And that's depicted by the radical and the dotted line. This deprotonation is followed by region stereospecific oxygen insertion at C5-alpha, which I'm going to draw for you right now. This yields taxodienol. Studies show that the hydroxylation step of Taxol biosynthesis is a slow step, or the rate determining step, in comparison to the following oxygenation step.
Taxol biosynthesis is significantly regulated by jasminic acid, JA. It's one of the most important indigenous hormones in land plants. However, the JA-inducing mechanism remains poorly understood. This is the current research of what they believe JA does. So the current research suggests that nearly all known Taxol biosynthesis genes are upregulated by JA and its derivatives, such as the TASY or TASI, which is the taxodiene synthase. Also the DBAT and the T7H. So we already went through taxodiene synthase in the two slides above, but structurally it proposes some interesting features that JA signals affect. So looking at this figure, we see the TCERF12 and the TCERF15. So the TCERF12 is a negative regulator, while the TCERF15 is a positive regulator. So in the JA signal transduction, the TASI gene, these two, TCERF12 and TCERF15, bind to the GCC box here, which then binds to the TASI promoter. And depending on which one is binding, it either inhibits or activates this TASI, which, as we reviewed in the last few slides, TASI is the first committed step, so it is obviously an important step to this synthesis. So another way we can put this is that taxodiene synthase, its promoter, contains an area which this GCC box binds, and it's found that this GCC box responds to JA signal transduction via the TCERF12 and the TCERF15.
As much of the Taxol synthesis is still under discovery, much of its regulation is still undergoing research as well. Two linked regulators are taxoid 13-alpha hydroxylase and 10-beta hydroxylase, and they are examples of regulators currently being researched. These regulators suggest bifurcating pathways early in Taxol biosynthesis. They take taxodiene 5-alpha, and depending on the R group on the 5-alpha oxygen, they lead down different pathways. So right there, it can either be a hydrogen, which turns into an alcohol, or an acetyl group, and that is indicated right there on the 5-alpha position. The 10-beta hydroxylase then further reacts to form a higher proportion of alternative products than Taxol, as you can see further down on that pathway. The significance of these results are not yet fully known, but suggest that these bifurcating pathways compete with routes that lead to other taxoid derivatives which can downregulate the synthesis of Taxol. All of our resources were obtained by peer-reviewed journals. Our data was found from research conducted within the last 15 years so as to provide the most current information on Taxol. Each slide directly links to the corresponding resources we used for the research shown and dictated for each section.